Well, thank you, Peter, and it's great to see some folks in person. I'm really glad to be here. I am a professor in the Department of Plant-microbe biology here at the University of Minnesota, but I'm also the scientific advisor for the Minnesota Mycological Society. And so one of the things that I've been, able to do and tried to help with is often giving a lecture of something on fungi, and most of the time the lectures are kind of topical about various aspects of maybe fungal diversity or maybe different ways that we're thinking about fungi. But I decided this time I'd actually talk a little bit more about some of the research that's happened in my lab and tell you a little bit about, as Peter said, the links between fungi and the carbon cycle, but have it really be sort of a Minnesota-focused talk. So I'm going to go ahead and share screen, and hopefully that's going to cooperate. So does that work for folks in terms of being able to see that version of it? Just wondering if that... and is it showing the extra slides that are in there as well? That's what it ends up looking like. Okay, so I think what I'm going to do, I'm just going to go ahead and I am just going to go through it in the way that I had it done before. So stick with us for just a second online. I think I am just going to, I think I'm going to go like that. That's just going to be as good as any way to do it. And I will stand here and try my best to look here and look at you, and then I can point up at the screen. So, great. Okay. So I'm going to start off this talk by actually giving some acknowledgments to the folks who did a lot of this science, and I'll talk about that picture in just a minute. But I've been really fortunate to work with a number of people here at the university who are at different stages of their career. So I've had some folks that have gotten their Ph.D.s and have worked in my lab as postdocs, both Chris Fernandez and Francois Maillard. Two really outstanding scientists who have done work that I'll talk some about today. And then I've had students, both undergraduate students as well as graduate students, that have participated in this project. And these are the folks that are ones that actually did a lot of the science that I'll talk about, although I've been lucky to work with many others as well. Fortunately, we've gotten some funding from the National Science Foundation. We've also gotten some funding from some internal sources at the University of Minnesota. And then I'd also like to acknowledge that a lot of the research that we have done has been on the ancestral lands of the Dakota, and particularly the Anishinaabe people. So this is work that is based up in the North Metro, mostly at the Cedar Creek Ecosystem Science Reserve in East Bethel. This is a picture that is actually from the top floor of the Biological Sciences building here at the University of Minnesota. I looked out my window. This was my first winter in Minnesota back in 2013, and this little mushroom popped out in the snow. In fact, two of those little mushrooms popped out in the snow, and that one made me smile. So we've always had that as our sort of lab motto, that we can still do mycology even when it's very cold. Okay, so the outline for today's talk is going to look like this. So first of all, I want to talk to you mostly about this kind of larger scale concept of linking climate change, thinking about carbon and soil and where fungi fit into that matrix. So I'm going to start there, and I'm going to talk really broadly about how those three things link together. After that, what I want to do is I actually want to zoom in and talk about fungi, and I guess I should say, spoiler for those folks that are online as well as here, I'm not going to show a lot of pretty pictures of mushrooms, but I am going to talk a lot about fungi. So I'm going to talk a lot about what fungi look like below ground when they aren't making the big, beautiful mushrooms that so many of us really like to look at, collect, and even eat, but play a really, really important role in the movement of carbon and nutrients through our ecosystems. So I'll explain more of these terms as I get further into the talk, but I want to talk about the decomposition of fungi, actually. And I can say that we've got some neat studies looking at who those decomposers are, but in this case, I'm really just going to focus on how fungal bodies, after they die and senesce, become part of the carbon that is in the soil and ultimately in the carbon in other atmospheric and, say, terrestrial pools. And so the lens that I want to focus on in the specific research that we've done has looked at basically 3 different factors. We've done a lot of work on actual other factors, but these are some fun ones that I think are good stories that highlight some aspects of the fungi themselves. So what are sort of intrinsic traits or properties that these fungi have and how that might influence the way in which they decompose. And so I'm going to talk a little bit about some biochemical traits and then a little bit about some morphological traits and some kind of fun experimental manipulations that we've been able to do to trick our fungi into making it so that we can make these kinds of comparisons. And then I'll talk about some experimental work that we've done thinking about where in soil fungi are, and does that matter for the way that they basically decompose. So that's the roadmap. And so to start, I actually want to start here. So this is a figure that is probably something that I'm guessing many of you have seen before. If you haven't, what you're looking at is basically the change in carbon dioxide concentration in our atmosphere since we really started making close and careful measures of it. And so this was a project that was started actually back in Hawaii in the 1950s on one of the high mountains in Hawaii, and careful atmospheric measurements were made. And what we see is this blue sort of saw that goes up and up and up and up and up. And that saw basically goes back and forth because it's in the northern hemisphere. And so when there are leaves on the trees, there's actually less carbon dioxide in the atmosphere because so much of it is being fixed and turned into sugars for those trees. And then when those leaves go away, basically the amount of carbon dioxide goes back up. But the inputs of carbon dioxide are increasing clearly over time. And then what you've got in addition to this is that red squiggly line. And that red squiggly line is showing you what the temperature in degrees Celsius has changed from what the average 20th century temperature was. And so what I think this graph really does is it highlights the fact that there is a pretty close link between the amount of carbon dioxide that is in our atmosphere and the amount of change in temperature that we are experiencing over the same time period. So with that in mind, thinking about carbon and thinking about carbon in our atmosphere, I think it's helpful to really kind of take a full step into where we would find basically carbon throughout our ecosystems and terrestrial ecosystems where we find lots and lots of our fungal friends. So this is a picture that is just a schematic trying to give you a sense that there are many different places, and what I had just talked about in that graph is where you find carbon in the atmosphere. And so that number is, so that's 589 petagrams worth of carbon. So if you're actually to take all the carbon in the air, in all the different molecules, and weigh it, that's what it would weigh. And so I find that a number that really has no particular value to me until I start to think about it in units of things that we might know the weight of a little bit more. And so I think if we do some conversions, maybe some of you think in grams. And so if you think in grams, this is still a really, really big number. 10 to the 15th is what 1 petagram is. And so we're talking hundreds of these things. If you're more comfortable in the English measurement system relative to the metric system here, a gram relative to pounds, right? It's a small amount of pounds in this case. But when you look at this, again, exponential number, and you think about how big that is, and you think about some other version of weight that you can come up with, if you do the back of the envelope math on this, 1 petagram alone is 150 million elephants. And so 589 petagrams is...
I know how much elephants.
You got that, right. Okay, so Peter's, yeah, we've got this set. So we now have a number that we can hold on to, okay? So we're talking really big numbers. But to me, what's, I think, important is basically thinking about where these different, again, sort of pools are that the carbon is in our ecosystems. And so we've got lots of it in our atmosphere. We've got about a comparable amount of it in the vegetation, okay? So that's, where we think, of course, wood, leaves, other, again, organic materials. That makes up a significant amount of the carbon on this planet. But the soil is actually the place where about twice as much carbon. So almost, put the atmosphere and the vegetation combined, and that's what you're starting to talk about in soil. And so when we think about soil as a basically reservoir for carbon, it really becomes the place where when we're thinking about climate change, or thinking about changing temperature and more or less carbon dioxide in our atmosphere, a lot of what happens in soil will change the amount of carbon that ends up in our atmosphere. So to think about how carbon moves in and out of soil, there's really kind of two basic processes. And so the arrow going in is productivity. And so for the most part, that is all basically plant derived. And so you've got this magic photosynthetic process where carbon dioxide is converted into sugars. It stays in the bodies of the plants, or it's pumped below ground. As the plants senesce, they add in basically their dead bodies, and this is really sort of the carbon pump into soil. As the process of decomposition is happening, right, these organisms are decomposing, breaking down organic matter, converting it into some aspects of their own bodies, but in the process, they are also respiring. So we respire, of course. We breathe out CO2 as we burn basically glucose and other sugars in our own body. And so that is in the process of decomposition, we have CO2 going back out into the atmosphere, and there are other carbon forms that are produced through decomposition as well. But so that's kind of how we think about carbon and how it moves around in terrestrial ecosystems. So this is basically the same plot. And what we're looking at again is the movement of, in this case, sunlight and CO2 into photosynthesis. It moves to the trees, and then it moves down. And here we start to see some of our fungal friends. And so there are inputs into the soil of carbon, and a lot of this is then fed directly to fungi that are helping plants get the nutrients and water and other resources out of soil. So these are the mycorrhizal fungi. These are the classic sort of plant helpers. They do a lot to basically allow plants to go after organic forms of various nutrients, as well as gather other resources, get protected from heavy metals, etc. And then there are the decomposer fungi, right? And so these are the ones that we think of that really basically live off of the dead organic material that's in soil, and through their own, in this case, processing of that material, they produce CO2. And so again, we've got this cycle of carbon moving through terrestrial ecosystems. Yes?
Question, I was going to save it until later, but if you don't mind now.
Absolutely.
Do you know how much carbon there is in the oceans? And Jean was saying most of the oxygen is produced by plankton, so you got lots when you're saying that it's the air, the soil.
Yeah, I mean, when we think about basically how much carbon is in our entire sort of planet, the vast majority of it is down in sedimentary rock. I'm thinking about sort of the active cycling carbon pool, but it's a great question, and yes, the number in oceans is much bigger than the number in terrestrial environments. So you can take those numbers and you can add to them. That said, even though oceans are vast and they're huge, there are many that have relatively low productivity. So I don't have an off-the-cuff number for what that is, but I do know that it is a very large number as well. Yeah, thanks and by all means, Kathy, jump in. I'm happy to answer questions along the way. Don't know if the question, could they hear you on that, or should I repeat the question? I can repeat the question. Yeah, I guess, the question was how much carbon in terms of petagrams is present in oceans, and that was what I was answering. Okay, so this is the carbon cycle as we sort of currently think about it, and we've got these distinct roles for these two groups of fungi, and so we think that the mycorrhizal fungi, again, are helping trees grow, and then we think of the decomposers as the real active respirers. That said, as mycorrhizal fungi go out and forage and live and do what they do, they also, of course, produce CO2, but they may be, in this case, not necessarily the dominant cyclers of organic material in this sort of classic view of how we think about these ecosystems. So we usually think the saprotrophs are the ones that are really involved in basically returning the carbon back into the atmosphere as CO2, and we think of the mycorrhizal fungi as helping basically get the nutrients that then allow the trees to photosynthesize, and so supporting that productivity. The thing that I want to talk about tonight, although I won't really sort of mention, is that there's sort of this exciting evolving perspective. We're learning more about fungi, and we're learning that fungi are really actually quite interesting with their own interactions, driving aspects of this carbon cycling. And so we're learning more that mycorrhizal fungi, in addition to helping the trees grow, have interactions with these saprotrophic fungi, and actually some of those interactions slow down the decomposition that happens from, in this case, organic matter in soil. And if it slows down the decomposition, that slows the amount of, in this case, CO2 that ends up back in our atmosphere. I'm not going to spend a lot of time talking about that, but I'm happy to answer questions. This is a topic that we've thought about and have some active research going on in the lab in Minnesota. What I want to spend more time talking about is this idea. So there was a paper, and there are multiples of these, but Science is sort of our top journal, and it was a neat study that was in a boreal forest, actually in Sweden, and what they found was that a lot of the soil, when they started to look at the carbon in soil, where it came from, it actually didn't come from plant material per se, and particularly above ground plant material. A lot of it came from below ground sources, so roots, as well as the fungi on their roots. And the numbers that they give for this are actually quite large. And so thinking that maybe something like half to 2/3 of the soil organic carbon in many, in this case, forests, but this is an example of a boreal forest, and potentially, hopefully, in our case, this translates into temperate forests as well. A lot of this comes from the fungi and themselves as hyphae. So to me, that's really exciting because it's this additional perspective that we have to add into thinking about when fungi are alive, what are they doing, but they themselves are a major input of carbon into soils. And just to sort of give another example of what that looks like, there was a recent review that looked at this topic, and what they found was that it turns out that microbes, whether it's fungi or bacteria, are a big part of the soil organic carbon across lots of different ecosystems. So we think of forests, and that's often, again, where we find lots of our fleshy fungi. Their contributions there in terms of the amount of soil organic carbon are maybe something like 1/3 in those ecosystems, but of that microbial fraction, a lot of that is coming from fungi. So you can see this red part of the pie chart suggests that of the microbes that are these big contributors to soil organic carbon, again, a lot of it is the fungi. And this holds across different ecosystems. You can see the numbers are a little bit bigger in, say, grassland ecosystems. If we move into, say, agricultural settings or cropland systems, again, the contributions from microbes is significant. Half of the carbon that's sticking into soil and staying there for a long time is coming from microbes, not just from plants. And so to me, this really underscores an opportunity to think about what role does fungal dead bodies, in this case, what we think of and what we call necromass, contribute to, in this case, the storage of carbon potentially below ground. So that's the sort of hopefully big picture combination of where we are thinking about climate change, increasing temperatures, where carbon is in our ecosystems, and how fungi, through their activities, but also through their own dead bodies, may contribute to the movement of carbon and potentially storage below ground. All right, so here's the sort of research side of what we're going to talk about. So got a slide here. So my lab is really excited about those, particularly mycorrhizal fungi as key symbionts of forest trees, but we've gotten increasingly interested in this idea that as the fungi, in this case, senesce, their bodies turn into this soil organic carbon. So that's the picture here. And that process ultimately is sort of as I said, decomposition. You're either going to end up as stored carbon or you're going to end up as CO2 at the end of that cycle. So in terms of how we focus today, these are the 3 areas that I want to focus on. So mycorrhizal fungi and their dead bodies, how do these different factors contribute? Biochemistry, morphology, and location. So I'll start with biochemistry. So the biochemical side of this is actually, to me, really interesting because it involves lots of different molecules. But one of the molecules we've been really excited about studying in fungi, that fungi have just like we as humans and actually other organisms have as well, is this particular molecule that is called melanin. So you can see melanin here is actually part of the cell wall for many fungi. It's an important, in this case, molecule that is actually very chemically complex and so hard to break down. And so it also is one that adds some rigidity to the cell wall. And so many fungal pathogens produce melanin as a way to stiffen their cells before they poke into the host that they are, in this case, pathogens of. But it turns out in soil, there's maybe some other additional sort of roles that melanin may play in terms of helping with things like drought stress. And if we look at a lot of the fungi that we find in soil, many of them have melanin in their cell walls. So this seems like an important chemical for us to think about as a trait that many fungi have. And so you can see what this actually looks like here. So you can see that fungi, of course, vary a lot in their chemical composition, and that melanin or that molecule, that pigment really changes. So we've got more and more, the darker it is, the more melanin that's present here across these different species. The issue with that comparison, right, is that yes, melanin is changing across those species, but so are lots of other things, right? There is other aspects of the chemistry of these species that make them quite unique and individual. And so to compare these species, ultimately what would be really nice is to have the same species that we could just change the melanin content of. So science is actually, a process that involves a number of relatively unexpected findings. This was, in fact, an unexpected finding in our lab. We kind of lucked out. And so we had an undergrad who was working in the lab and was quickly putting media that we were growing fungi in these flasks into, and accidentally varied the amounts of media that were present in each of those flasks. And when that happened, it turned out that that really changed the amount of melanin that was produced by this single fungus. And so we have a picture here of what that looks like. So you're looking at these nice white sort of fluffy fungal colonies that are growing in the sort of deeper, in this case, the more full flasks. And then in the lower flasks, you can see that when they're actually exposed to much more oxygen, there's a lot more production of melanin. So in this case, the production of melanin involves basically, it's a biochemical pathway where one of the enzymes is really oxygen, in this case, sensitive and needs oxygen to go through its enzymatic process. And so when it has lots of oxygen, it produces that melanin. When there's less oxygen around, it actually doesn't actually melanize the fungus very well at all. So this fungus gave us a chance to kind of hold species constant. And it's a process that we really, done it a number of times now. It's quite consistent. We can see that the colonies, in this case, on the right side of the slide for me anyways, are much darker than the colonies on the white side. And so we decided to use this as a study system to investigate this question. And so we were interested in thinking about melanin and melanin concentrations, and we measured this. So there's some other ways to measure this, aside from just visually saying, wow, this dark one is quite melanized, this light one not so much. And we can see that there's really a big difference between these. And so we feel confident that melanin is varying between these, not just on the outside, but also across the entire mycelium. So this is work that was done by one of those scientists in my lab, a guy named Chris Fernandez. And Chris decided to kind of take this question and put this as an experiment where he'd actually looked across different species, but not within a species, into the ecosystem to see how melanin influenced decomposition. I should say that this fungus, again, unfortunately, isn't a big mushroom producer. It is a common mycorrhizal associate, but it is an ascomycete that is, again, present in soils. It forms mycorrhizal associations with lots of forest trees, but also ericaceous shrubs, but it doesn't make big showy mushrooms. And so we don't get to study it and enjoy it from that perspective. That said, we've cultured it and we can grow it, and so it's become one that we've worked on quite a bit. So this is a picture of Cedar Creek. Cedar Creek is actually quite famous here at the University of Minnesota because it's the home of our one of our research outreach centers at the university, and it's the home of the Cedar Creek Ecosystem Science Reserve. There's been lots and lots of studies of all kinds of ecology in this reserve. It's located about 45 minutes north of the Twin Cities. And so what we decided to do is actually go out to Cedar Creek and work in a couple of different forests that were basically adjacent to each other. And so we could hold things like the climatic conditions, basically the same between these two forests. We could say the original soil from which these forests started on was relatively comparable. So we could ask how kind of generalized this pattern was. So if we decompose this mycorrhizal necromass in these different forests, we get a similar result, we get a different result. And so that was why Chris and I decided to work in these two different types of forests. So we did that, and I'll show you sort of what this looks like. This was an experiment where we took these little chunks of, in this case, dead mycorrhizal fungi. So we'd grow them up and then we would dry them out. And I can promise you that these don't come back to life. So we don't have any zombie fungi here that are growing in our bags. We just have the dead fungal material. And these are really basically like tea bags. They keep plant roots out, but we use these because we want to take that fungal material, put it out with a known weight, and then we want to bring it back to the lab after it's decomposed and weigh it again to figure out kind of how much has decomposed. So you can see what this looks like. We've got these black and we've got these white different, in this case, necromasses that we are putting out. And we harvested them over a series of times. So we wanted to kind of let decomposition run its course. And to do that, we waited 1 month, 2 months, and 3 months, and then individually harvested these bags and brought them back to the lab. So this is what they look like when you zoom in on these bags, and you can see that there are lots of fungal hyphae of other fungi that have colonized these bags. So we're learning that the decomposers of fungi are often, in this case, fungi, although bacteria are also quite important. There's some interesting patterns that are starting to emerge there, but definitely fungi really like to decompose many of their fellow other fungi. All right, so I'm going to show you a graph here of what this set of results, this first set of results looks like. And so what we've got on the x-axis is we've got time. And so we're looking at the different time periods where we sampled these, in this case, bags full of dead fungi. And then what we've got on the y-axis is the amount of mass remaining, right? So if you have a lot of mass remaining, that means there was very little decomposition. Whereas if you're way down at 0, that means everything, all the fungus that we put in the bag is gone, okay? And then we've got colors to our bars. And so the dark bars are the ones that were highly melanized, in this case, dead fungi, versus the light bars are ones that didn't have the melanin or the very low levels of melanin in there, in this case, their necromass. So what we can see if we look at this is that there's really a pretty strong and clear trend, right? Where the black bars are higher consistently, regardless of whether you're in the oak forest or whether you're in the pine forest, that there's a lot more mass remaining there, right? So that suggests that the decomposition is much slower for the dark or the melanized fungal necromass than it is for the non-melanized necromass. And so we can run some additional analyses that really allow us to test this statistically. It's one of these that really passes the eye test, in my opinion. We don't need fancy statistics to tell us that there's this difference and that, again, there's a change over time. And so there's less mass remaining. That's what this is saying. Melanization matters. So the black necromass decomposes more slowly than the light or the non-melanized necromass. There is a little bit of a side effect. And so what we see is that overall things are going a little bit faster in the pine forest than they are in the oak forest. And there's a little bit of a kind of pattern where the difference between the black bars and the gray bars is a little bigger early in the experiment and a little smaller later in the experiment. And so that's kind of walking you through what that is there. But for the sake of what we're talking about today, the main take-home message here is that we're seeing melanin is really looking like this important, in this case, molecule that's slowing down decomposition of dead fungi. So this is really consistent with what Chris had found before across different fungal species, but now we have this capacity to say it's not just a pattern that we think is melanin, but we're not sure. We feel much more confident with these two studies together that melanin is this slowing down of decomposition. So if melanin slows down decomposition, and if we're going to think about that from again, ecosystem perspective, that's going to mean that more of the carbon's going to stick around in soil and not end up back in our atmosphere. So if we want to think about sort of climate change and we want to think about ways to slow down basically the inputs of carbon out of soil, melanin might be actually really quite important. So that's the first story that I wanted to tell you guys about from research we've done. And so that covers the sort of biochemistry side. So let's move on to this next term, which is morphology. And so this was a kind of fun experiment that's got a little bit of fungal biology underneath it. So I'm going to start here first, and then I'll work into the experiment. And so if you were to go into a forest and you were to, in this case, look closely at the roots of many of the trees in the forest, particularly oaks and pines here in Minnesota, you'd see that they have these little fungal socks that vary in color and they vary in size and shape. And these are the mycorrhizal fungi. And the way these grow out from the roots that they are on top of varies a lot. And so you can see we've got versions of it that are sort of, in this case, brown with very little sort of emanating mycelium. We've got these sort of black ones that have maybe a longer, sort of darker, more bristly-like mycelium. We've got these white ones here that have these long kind of cord-like structures, and then they have this much more sort of even or diffuse mycelium. So if we break this down, we can show that there's sort of this pattern of kind of two major forms. And this is true of most fungi, regardless of whether it's mycorrhizal fungi or saprotrophic fungi. The growth of mycelium is often really quite even. So individual hypha of a fungus, when it grows out in a way that is often quite radial and quite evenly distributed. And so that growth is actually really a good strategy when you want to put yourself right next to what you're eating, which is what fungi do, right? So unlike us, they don't have internal digestion. They have to put their bodies next to what they're eating. They put those enzymes out into the substrate, and then they slurp up what has decomposed. And so they want to be right on the surfaces where they're growing. The issue with that is, think about soil. Soil is, really patchy and heterogeneous. There's lots of little pores. There are some resources over here. There's resources elsewhere. And so evenly growing across the surface is maybe not as efficient, right, in terms of being able to target the patchiness that's present in soil for the different resources that fungi are interested in growing on. So in this case, we've got a second version of growth, and that's actually what that cord-like structure is here. And so these cord-like structures, they've got different names: cords, strands, rhizomorphs, so kind of root-like structures. They do a good job of going around and moving fungi and finding the location where there are lots of resources, and then oftentimes from there, the fungus will grow out in a more sort of diffuse mycelium that again helps localize the resources that are available. And so a rhizomorph is what we refer to as this mycelial organ. So it's basically a tube. It's sort of like a plumbing structure for fungi, where it's fused a bunch of hyphae together, and there is a opening in the middle. So it really is sort of tube-like that makes it so that lots of resources can move from one location in the fungal body to another. So this helps basically, again, fungi connect patchy resources that they're looking for on the landscapes that they're living in. So this is an example of a pine seedling. This is some work that we did in the lab, and this is work that was led by Francois Maillard. And so what Francois did was he had our fungus, this mycorrhizal fungus, and he got it to grow on its own, and then he resynthesized the symbiosis with these little pine seedlings. And so we can see these root tips. So these are, again, the brown fungal socks this fungus makes this tree wear when they're in symbiosis. And here's an example of what it looks like. You can see the hyphae that are there. If you were to take a cross section, you'd actually see some of these mycorrhizal sort of structures where the fungus is growing and wrapping around the tree cells. That's where the nutrients and the carbon are being exchanged. But if you look closely at what's going on in the plate here, you can see that there are these cord-like structures, or these rhizomorphs. And if you collect these out in nature, you can also see a lot of these rhizomorphs that are present on these root tips as well. So in terms of thinking about mycorrhizal fungi, this is a trait that we're really kind of interested in and exciting about because it looks like it's really important from a functional perspective. And yet we don't really understand how, when these fungi die and they produce these structures, does that matter at all for this process, like how fast it decomposes? So the issue, unfortunately for us, is that we tried to think about how to do this using mycorrhizal fungi as our study system, and we just could not come up with a way to scale this up and generate enough of these rhizomorphs. And so luckily, we were able to use a different fungal species that I think many of you might actually know as one that could produce these structures, and then we could use this as an analog to think about how it might work for mycorrhizal fungi as well. So we used the honey mushroom, which is a beautiful mushroom that we find a lot of in Minnesota forests in the fall. So Armillaria mellia, although there's actually a lot of Armillaria species. We went out to our location, and actually before I go there, so many of us think of this as the sort of exciting look for the honey mushroom. For me, a second part of this really interesting look of this fungus, it's also known as the shoestring fungus. And so if you have had any time walking around in forests and you've either stepped on a rotten log or you've picked off the bark of logs, you'll often see these really long black cords that are formed. And that is Armillaria, and growing in these long rhizomorphic structures that eventually, when they find a place where they want to grow and they grow out, they have that diffuse mycelia. So you can see these sort of white, more diffuse mycelia connected to these long black cords that are really strong and actually are quite almost shoestring-like. And so hence the other name for this fungus. So I got to work with a really talented student at the time, Amanda Shotano, who was an undergrad here at the University of Minnesota. We went out to Cedar Creek. We collected this actual specimen right here. So this is the honey mushroom that we worked with. We brought it back and we got it into culture. And interestingly, the honey mushroom does some funny things when you grow it in culture. And so if you look at this, what you can see is that you're seeing the outline of where the fungus is growing, and then you can see these weird, almost antlers that are popping out. And those are the rhizomorphs that it produces in culture. And if you're looking really closely, I'm sorry, it's quite pixelated. It turns out that these fungi, when you add a little small amount of alcohol into the culture conditions, produce much more rhizomorphs than when you don't add any alcohol in. This was a discovery that Amanda found in the literature from the 1950s. Why and how that started, I think, might have, again, been one of those happy scientific accidents, but it turned out for us to work out really well as a way to either have rhizomorphs or not. And so what Amanda did was she did this kind of cool setup where to harvest these samples, we could grow the fungus in liquid media that was glass beads. So it's filled with glass beads. So you have the fungus up on the surface, and so it's able to have plenty of oxygen. It's got the media full of nutrients, and it turns out if you add the alcohol in the beads, the fungus loves to, I don't know what it is about the sort of tactile surfaces, produce rhizomorphs. And so I've just taken one of these, or Amanda took one of these and flipped it over. It kind of looks like a jellyfish, or like a sort of amazing what the morphology is. But we could give it a haircut, and so we could trim off all its little rhizomorphs, and we could compare that to mycelium that was grown. It's the exact same strain. You can see here it's got that diffuse kind of blob-like morphology that is sort of non-rhizomorphic. So we had our system set up to test this question, where we went back to the exact same forest because we thought, you know what, we've had some luck looking in this forest for this question. What if we kind of go back out with now a new trait, but in the same ecosystem? And so we went out and we did the same experiments. Again, this is not a fungus that has come back to life. This is actually fungi that have gone in and colonized. You can actually see some of this brown mycelium here that's actively colonizing these rhizomorphs that are present in here. We did it over a little bit sort of different schedule than we did in that first experiment, but basically it was another 3-week experiment, a different summer, and then we went back to the lab and we assessed how much it weighed. So, second round of results for you. So this is a plot of what these results look like. So I want to walk you through what's here. And so what you can see is, again, we've got our x-axis is time. And so at the origin, we're at 0 when we started the experiment, and then we have increasing amounts of time. The y-axis is, again, the amount of fungus that we have left. And then the two different morphologies are the black lines or the blue lines, and the dotting on the lines versus the solid lines is which forest type it was in. And so what you can, I think, pretty clearly see here is that there's a big difference in the amount of mass loss or the amount of mass remaining, depending on how you measure that, where the rhizomorphic, in this case, versions of our necromass were much slower to decompose over this 12-week period, where we only lost about half of the biomass or the amount of weight from those, and then we lost quite a lot more from the diffuse version of that. So if we think about what this means, it really suggests to us pretty strongly that morphology is also a variable that we should pay attention to in terms of changing the amount of decomposition we see. And so in these more rhizomorphic forms, there's much, much slower decomposition. And the half-life, if you do these calculations, is basically much longer. So if we have rhizomorphic mycelium, it's going to stick around in soil longer than if we have just diffuse mycelium. So those are two of the traits that we've looked at. So what I want to do now is give you the last version of what we've looked at here, and that is looking at, in this case, the location of where these fungi are. And so we'll go there. So Chris, when he was in the lab, did this really kind of cool literature summary and kind of thought about sort of what are the variables that are important to think about in soil in terms of carbon cycling and fungi, and particularly mycorrhizal fungi. We've already talked about this idea that traits matter, and so we talked about this idea of in this case, whether it's got melanin or not, and how important that is in terms of the chemical recalcitrance of that necromass material, and sort of what that means for soil organic carbon versus carbon dioxide and decomposition. I haven't talked a lot about this idea, but there's actually ways that dead fungal bodies might stick into and get protected from decomposition based on some of these traits. That also is potentially important. And then the fungi themselves, as they die, they may create these nets of various sort of intricacies, and that may also lead to aggregates of soil that can affect the amount of carbon that's there as well. So that was on that side, but we had this whole sort of other part that we were thinking about, and the idea was that the depth of where these fungi are, these mycorrhizal fungi occur at lots of different depths in soil, but we know that there are some differences in basically the abundance and the activities of decomposers, and we usually see a relationship where there's lots of activity of decomposers, there's less amounts of carbon around, and then as you go down and there's less and less of these decomposers, actually the amounts of carbon in soil increase. So with that in mind, I was really lucky to work with my current graduate student who I co-advised with another professor here at the University of Minnesota, Lang Delancey. Lang is a master of many things. He is probably the best morel hunter that we have in the lab, maybe because he's from Michigan, that may be part of it. But Lang also really likes to do all kinds of crafts. And so he and I decided, let's try this experiment where we actually make these ladders, these depth ladders, where we can put fungal material in at different depths to soil to say, okay, we're up at the surface, or we're sort of mid-level, or we're lower level. And so with this router tool and some time in Lang's home workshop, we built these out of these very hard sort of PVC, in this case plastics, and then we constructed these little, again, sort of tea bags full of dead fungi, and you can see what that looks like here. This is the traits that we worked with. We went with the very melanized version of that first fungus that I told you about, Millennium-Ices bicolor. We put this out in 2 locations out at Cedar Creek. One of those was the same as the first 2 stories that I told you about, but there was another one we were interested in sort of thinking about trying to replicate this across some different sites. we have these different depths. So this is where the fungus, the dead fungus, is actually located. So we have, in this case, 3 different depths in the soil, and then we had one sitting right up at the surface. We put this out over the summer, again, sort of the similar 3-month time period where we give enough time for things to decompose, but we can still measure it. And we, again, looked at what was remaining at the end. So Lang did this. It was kind of neat. This is a version of it that has been buried, and then he went back out, and this is what it looks like when he takes this out. So he's got a really fancy tool to recover this, and you'll see here what this looks like. And there they are. So that is the pine forest, and again, that's the ladder that we have of these decomposing little bags of fungi. So, okay, so just to tell you why we only focused on one version of the fungi rather than kind of multiple versions as we had in these other experiments, we see again and again in all these different studies that we've done this pattern, right, where we end up with a lot of early decomposition and then really kind of a stabilization of the amount of fungi that remain. And so this pattern was consistent enough, and ultimately this amount remaining probably has a lot to do with, again, how much carbon stays in soil as a fungus. And so we wanted to kind of focus on that end more so than we were interested in the sort of the early decomposition dynamics. And so that was kind of the impetus for us thinking about, if we focus on a later time point, we're really focusing on this slow decaying fungi, which to us is the important carbon contributor. And so we are back over here. And again, if you zoom in on kind of what we were originally thinking about with Chris in this commentary that we had written, and you sort of took these predictions and you mapped them out onto what they would look like in terms of their maybe the mass remaining patterns, you'd expect to probably see a graph that looks something like this, right? So where there is the most activity and the most abundance of decomposers, there's the least amount of mass remaining. And then as you go down with depth, right, into the soil, you'd have more and more mass remaining. So that was where Lang and I started with our predictions. So we did the experiment, and again, the other process in science that you learn is that you do lots of things, and you're sometimes surprised. Your predictions maybe don't always work out quite as cleanly as you might think. So this was sort of a puzzle to us. We were sort of interested. Maybe we see a little bit of a pattern over here in this case, maybe where there's some more decomposition, but it's definitely not really clear that as we're sort of going down, actually we're getting that sort of consistent pattern that we had expected before. We've been thinking about this data. We haven't really gotten a chance to kind of work our way all the way through it, but I think there are a number of reasons why we might have gotten this result. One is that, maybe we just didn't get down far enough into soil to actually get to where really there's not as much decomposer activity. And if that was the case and we were up in this more active zone, then it would probably make it such that potentially all the depths were more similar. So that's one variable, and actually working at Cedar Creek, it's an interesting, very sandy ecosystem, so it's a little bit different in that way, and maybe that's part of what happened. We may have missed some of the patterns. Maybe there was some interesting patterns early on that we didn't get a chance to catch by just focusing on that one later time point. But to me, I actually think this is probably, the fact that we focused on this really highly melanized necromass in the first place is that I think regardless of what depth you're at, it's hard to break this stuff down. It's chemically complex. It has these aromatic rings that just don't get easily broken down by most organisms. And so I think in that way, maybe part of the reason why we see the same pattern across depth is because we have this highly melanized fungus. So don't see anything in this case that really suggests location based on this data anyway is critical for thinking about decomposition patterns. Okay.
One of the questions from the chat was about the variable moisture levels by depth. in topsoil versus subsoil?
Yeah, that's a great question. So I don't know if folks heard that question, but it sounds like there's one in the chat, so you can see it there. So we did look across depth, and there was what you would expect for some amount of variation in soil moisture. And so there's more soil moisture at depth than there is up at the surface layers. That said, this is kind of, like I said, a weirdly sandy soil. And so the amount of soil moisture in this system is one that there's not a lot of retention of the moisture in the soil. And so we haven't spent a lot of time sort of digging through and trying to sort of put all those pieces together. But that's a good question. And the other question was about how much there's sort of a gradient between the topsoil and kind of the organic layer, and then maybe down in the more mineral soil. Again, kind of a unique property of Cedar Creek is that there's not really a well-developed sort of organic horizon. It really is quite sandy throughout that entire depth. And so in some ways, it allows us to look at depth kind of without those variables being quite as strong as they might be in other systems for changing amounts of decomposition. But certainly moisture matters, right? Fungi need moist conditions to grow. And so I think there's generally a correlation between more moisture, more decomposition. We're not really seeing, again, super strong patterns. Again, fungi need nutrients to grow as well, but you'd think they'd get a lot of that out of the necromass in addition to the surrounding soil. So I'm not sure they're nutrient limited in that system or how much the external conditions would matter. So that's there. So I guess I just want to conclude with a kind of a handful of messages here. So I hope what I've done in this talk is convince you that fungi are a big part of the soil carbon cycle, both when they're alive as decomposers as well as mycorrhizal symbionts, but also when they're dead. I feel like fungi are so notoriously known for being really important as the turnover of dead bodies, but we usually don't think of them as inputs in their own dead bodies being really important. And yet we're consistently seeing more and more that that's a big part of the carbon that sticks around in soil for long time periods. With the science and the results that we talked about in the studies today, I think we can comfortably say that there are things about fungi themselves, their own biology, whether it's their chemistry or whether it's the way they grow, their mycelial networks, that drive the decomposition rates that we see in their own dead bodies in soil. And ultimately, I think where we are interested in sort of thinking about this is in the context of this idea that if we can change or understand or in this way better control the sort of rates at which fungal necromass decomposes, that can ultimately be quite important potentially in the retention of carbon in soil, which if it stays in soil and not in our atmosphere is important for slowing down the rate at which our climate is warming. So I think that's basically it for what I wanted to cover. I think I'll just jump right down to this slide and say thank you for the in-person audience, and thanks for the folks who have joined online. Happy to answer any questions if anybody has any questions.
So
Ron's got a question. Great.
Determines the degree of melanization in
Mm-hmm.
these different species. And also, could you just put alcohol in the soil and increase the rhizomes? My thought exactly. Honestly had not thought of that, and so that's an interesting. Might have to run out to the corner store.
Bottle of whiskey,
Yep.
vodka, and
I think that's
sprinkle it on your.
Dry November. I can take all the booze and just put them out in the forest, and I'll be helping store more carbon. I think that ethanol one, they're sensitive. It actually, if you put too much ethanol in there, fungi don't really like it, and so that's why eventually you can suppress the amount of alcohol that accumulates in distilled liquors or in wine, etc. And so no, I don't think we can add alcohol to soils and magically generate more rhizomorphs and therefore more carbon in soils. But I like the idea. The other question is about the variation in melanin across species, and it's really widely variable. I mean, it's interesting. We can think of things like dead man's fingers, right? Xylaria, polymorpha, or that group. I mean, there are lots of these that are just dark and crusty and have all kinds of melanin in their cell walls. Camerops, Petersiae, the dog nose fungus, that's another one that's just super well melanized, and you can see there are a number of fungi. One of these mycorrhizal fungi produces highly melanized mycelium. Doesn't produce big fruit bodies in the soil at all, and so we don't see it. But it's a trait that, again, when they've done these comparisons and they've looked at a bunch of different ecosystems, and you harvest the soil and you float the hyphae out, and then you stain them and see how many of them are melanized versus not, you get a really surprising amount of taxa that have melanin in their cell walls. Like it is a significant amount. I mean, the idea that at least half or maybe 2/3 of those fungi, depending on what ecosystem you're in, have melanin, suggests it's a common trait. Like I said, it's got different ecological roles. And it's kind of interesting. I don't think that, melanin has the same role in the fungus as alive as it does when it's dead. And so it's kind of one of these cool kind of phenomena where there's this sort of afterlife effect, where, again, when fungi are alive, they often use melanin for dealing with, sort of potentially drought stress or helping them be better pathogens. Maybe there's some interesting things that happen with UV protection that are totally unlinked to then what happens in their role in carbon cycling afterwards. How much this changes depending on like environmental conditions. Can you get the same fungus and in one place it's dark and in one place it's light? I don't know. That's a question that we've been able to trick these fungi to do fun things in the lab, but I'm not really sure how much that varies in the same fungus, say, at different depths in soil. I could imagine that there are important potential oxygen gradients or otherwise that might also matter. And so, people have looked, and there tends to be more melanized fungi at depth than there is up high in soil. But that's a good question.
Melanomyces, is that an ectomycorrhizal?
It is ectomycorrhizal, and it also is, it forms mycorrhizal associations with ericoid mycorrhizal hosts. And so ericoid shrubs, and so, leatherleaf and all the kind of bog plants, a lot of these have that association as well. Mm-hmm. But it forms ectomycorrhizal root tips with pine trees and birches and some other hosts as well.
I don't have a question, it's just more of a comment.
Please,
about how when you put the samples, actually planter them.
Yeah. Hold on. Let me just go from, I'm going to stop sharing because I think there's anything else that's super important there. And
I'm unmuting to ask my question. Oops.
just quickly,
Perfect. Just that it seemed like all the samples you put in the soil, other mushrooms wanted to kind of attack them. So while you're measuring degradation, it seems like a part of the puzzle could be really complex if you could get the right succession of species where one's eating another, so that nothing's truly ever decomposing, it's feeding something else and getting, like you were saying, it's like if, I don't know which ones like to eat which other ones, but it's like if you could find out the ones that were, like to consume the other ones, that you keep it in that consuming process rather than the kind of exhaling process.
Okay, we're perfect. We have a little bit of feedback in the room, and so we're switching mics here. I love that question or that comment, Kathy. I think it's really insightful and really cool to think about kind of what role, in this case, the sort of feeding and movement of carbon right through these ecosystems and how that works. The real initial input and the fixing of carbon out of our atmosphere comes out of plants, and then that carbon ends up in the mycorrhizal fungi. So that's pretty close to the sort of original source of where that carbon is being moved into the ecosystem. But then from there, and we've done a lot of work to show who the fungi are that are eating other fungi, and there's kind of there's a couple of patterns that are fascinating. So I'll tell you one. is that Millenniumyces, this ericoid mycorrhizal fungus, when we've done some experiments in other ecosystems, we found if you put that out and you characterize who the fungi are as decomposers on, say, Millenniumyces versus a different fungus that you could put out, Millenniumyces is enriched by like a huge margin in other close members of that genus, but not the same species. So there's like almost a kinship, and it's not a cannibalism per se. It's not the same, but it's this weird thing where either there's like, they're better at chemically sensing closely related taxa, or they're better chemically set up to decompose themselves. So that's one sort of kind of interesting pattern that we've seen with that particular taxa. The other is that, I think your question is sort of how do you keep that carbon sort of cycling and staying below ground? It can stay in these loops for long periods of time in soil. Ultimately, if it ends up though as a dead fungus that sticks onto some soil mineral or some particle, it will then also stay there, and then it can get recycled and put back and recycled and put back. But there is fungi are as lots of things like bread in your fridge, or if you leave something out, there's just a whole pile of fast-growing molds that love to eat other fungi. And so we're learning a little bit by using some kind of carbon labeling techniques where you can grow up your fungus with carbon that you have that has a particular signature, and then you can watch it go into the ecosystem. So you can watch it go into these microbes, you can watch it dump out in the soil, or you can watch it go up in your atmosphere or stick around and that kind of thing. So we're hoping to continue to work on that more. And there's lots of other fungi in there. I'm pretty convinced that even though there's lots of other fungi decomposing that material, it's not like they grow into the bag and really change the weights. That's one thing that we've often gotten critiqued on, is this idea that, are your weights accurate? If anything, our weights are conservative, and yes, there's probably other fungal growth, but we don't see scenarios where fungi suddenly sort of, those bags anyway, gain weights. We just see consistent levels. Is actually the last thing I'll say is that there's sort of interesting things where we've also been able to show in various studies that mycorrhizal fungi, again, go after their own, in this case, necromass. We can find them in high abundance on the dead body. But again, they get their carbon from the trees. And so they're not interested in the carbon that's in the necromass. They're interested in other stuff like the nitrogen. And so there might actually be this kind of closed loop where the plant feeds the fungus carbon and the nutrients and other things that it puts in its body, and then other fungi can access more of that as they sort of give that back to their host.
Thank you.
Sure. I can't really keep an eye on the chat at the same time. I don't know. Is there? Okay.
Just a lot of people thanking you for the presentation, and they
cool.
found it very interesting.
Sure.
Gene Kramer says Dr. Ingham of the Soil Food Web would be interested. Thank you so much.
Yeah, I'm starting to think, so one of the fun things that we've also done is when we characterize the microbes that decompose the fungi, you can do that in lots of ways. We use it doing molecular techniques, so we can go after DNA, and we can basically go after groups that we know are in there. So we've gone after the other fungi, we've gone after the other bacteria. We've just started going after the rest of the kind of larger microeukaryotes, so things like protists or nematodes, and we're seeing that there's like a whole bunch of, for example, nematodes that eat fungi or bacteria that just are a big deal in these piles of dead fungal mycelium. I'm starting to kind of, in my mind, use the analogy of like a Serengeti watering hole, where it's like the water's there, that's the fungus, and then there's like all these other animals, and there's like crocodiles, and there's just a whole lot of things, and they're kind of these ephemeral resources that, again, pop up and go away on the landscape. But it is really a complex ecosystem that we're just starting to kind of pull at different pieces on. Well, thanks. I'm happy to answer questions. Yeah. You guys have my email. Stay in touch.